Good day folks, welcome to my channel. In this video, I'll be discussing about the free radical scavenger system. So here you can see the diagrammatic representation of the free radical scavenger system. So here the superoxide anion radicals, two superoxide anion radicals combined with two hydrogen ions, and this in the presence of the enzyme superoxide dismutase forms the hydrogen peroxide and oxygen. You know that this hydrogen peroxide formed, it is also considered as an example of free radical, though it is not highly reactive. In the presence of ferrous ion, it will be converted to the hydroxyl free radicals, which are highly reactive. So this peroxide ion form have to undergo various other steps to finally form water. So the next step that follows is the hydrogen peroxide for hydrogen peroxide form is converted to the water molecules, and this takes place with the help of the enzyme glutathione peroxidase. Now this glutathione peroxidase acts by converting the reduced form of glutathione to the oxidized form, and this conversion is reverted back by the help of the enzyme glutathione reductase, which maintains the glutathione in the reduced form so that further conversion of the hydrogen peroxide to water molecules are made possible. But for the following state to be maintained, energy is required in the form of NADPH, and this NADPH it is converted to NADP plus, and this NADPH it is obtained from the HMB shunt pathway. So the HMB shunt pathway acts by converting the glucose in the presence of the enzyme glucose 6-phosphate dehydrogenase to this energy molecules, that is NADPH. Now this is the basic structure of how it happens or how the free radical scavenging system works. Now let's see these enzymes one by one. So the first enzyme, which is a superoxide dismutase, so it is present in the mitochondria as well as in the cytoplasm. Now the superoxide dismutase, which is present in the mitochondria of the cell, is a manganese dependent enzyme, whereas the superoxide dismutase, which is present in the cytoplasm, is a copper zinc dependent enzyme. And the superoxide dismutase, it is denoted by capital letters SOD. Thus, SOD is a non-heme protein, and if there is a defect in the SOD gene, that will lead to the disease known as amyotrophic lateral sclerosis or Lou Gehrig's disease. Now moving to the next enzyme, which is a glutathione peroxidase. So it is a selenium dependent enzyme. As we've already mentioned, the main function of the glutathione reductase is to convert the oxidized form of glutathione to the reduced form so that further conversion of hydrogen peroxide to the water molecules are made possible. So this utilizes the energy in the form of NADPH, and in the common kind of disorder in which there is deficiency of the glucose 6-phosphate dehydrogenase enzyme, the conversion of the oxidized form of glutathione to the reduced form of glutathione are not made possible, and thus this step, that is a conversion of the hydrogen peroxide to water molecules, does not take place. As a result, there will be accumulation of the hydrogen peroxide, and this especially causes hemolysis of the RBC membrane. And especially when oxidizing agents are administered in the body, such as, and this kind of disorder, it is known as drug induced hemolytic anemia, because hemolysis result. Now another important enzyme that we have to discuss in this concern is the catalase enzyme. The catalase enzyme, it is present in the peroxisome. As you may know, it is a marker enzyme for peroxisome, and this catalase, it acts when the H2O2 is generated in large quantities. So usually for the conversion of the hydrogen peroxide to the water molecules, the enzyme glutathione peroxidase is the one which helps in this process. But when there is an increase in the concentration of hydrogen peroxide which is formed in the body, the enzyme catalase works to convert the hydrogen peroxide to water molecules and oxygen molecules, this is the following step that takes place. When there is excess accumulation of the H2O2 in the presence of the ferrous ions, it will be converted to the ferric ions, and this ferric ions and hydroxyl ions, and this hydroxyl ions they are hydroxy-free radicals. They are basically create high havoc to the living tissues. Now let's take a quick look into polyphenols. So polyphenols, the consumption of polyphenol-rich fruits, vegetables and beverages is beneficial to human health. So the dietary polyphenols, they occur in fruits, vegetables, wine, tea and chocolate, and they contain the components such as flavones, isoflavones, flavonols, catechins and phenolic acid. So these polyphenols, they have the antioxidant property, which and also the anti-apoptosis property, anti-aging, anti-carcinogenic, anti-inflammatory and anti-atherosclerotic effect. So this antioxidant property, it helps in the free radical scavenging system. That's it for the video. If you honestly did understand the topic, do like, share and subscribe. Thank you.